Now in part 2 of glycosaminoglycans and mucopolysaccharides, we will be discussing about the mucopolysaccharidosis. What is this, sir? In simpler words, see, we have lysosomes with lot of hydrolases, right? So with lot of hydrolases, so lysosomes contains hydrolases. Along with that, we have glycosaminoglycans, and because of this hydrolases present in the lysosomes, our glycosaminoglycans will be degraded. Okay, so what I want to know is the degradation of the glycosaminoglycans will be taking place in where? Lysosomes, right? So because of degradation, we call it as mucopolysaccharidosis. Very simple. So it is a progressive, that is the hereditary progressive disorder, right, which is associated with the mutation of genes, and that will code for lysosomal enzyme. So because of the mutation of genes which is coding for particular lysosomal enzymes, and because of this hydrolases, our mucopolysaccharides will be degraded. Very simple. Okay, so there is intralysosomal accumulation of mucopolysaccharides, right? So there is when this they are not degraded, when this is not degraded, then there will be intralysosomal accumulation of mucopolysaccharides. Am I right? See, mucopolysaccharidosis means what? When this degradation is not taking place, then it is mucopolysaccharidosis. If this is not taking place, what will happen to the glycosaminoglycans in the lysosomes? It will accumulate. When that accumulates, okay, so we call it intralysosomal accumulation, right? And it is a lysosomal storage disorder. So no, our mucopolysaccharides, it is storing in the lysosomes. Very simple. You understand the concept? Okay. So I'll just repeat again. Very simple. We have the hydrolase to degrade our mucopolysaccharides. When this degradation is defective, it is not taking place, then our mucopolysaccharides will be accumulating in the lysosomes. If that accumulates, that is called as mucopolysaccharidosis. Very simple. Okay. Now, if that accumulates, if these mucopolysaccharides accumulates, then what are the features present in the patients we are going to see? Very simple. That is, we have the coarse facial features, that is frontal bossing and depressed nasal bridge. There is large tongue, intellectual disability. If you see, okay, I am putting a box in such a way that it is seen in some of the mucopolysaccharides, some of the mucopolysaccharidosis. Okay, and also there will be corneal clouding, copious nasal discharge, visceromegaly. That visceromegaly means what? Hepato as well as splenomegaly, which is leading to protruding abdomen. There will be protruded abdomen with hepato and splenomegaly. That's called as visceromegaly. Okay. Next, there will be umbilical and inguinal hernia, and there will be skeletal dysplasia, which means in the X-ray, if you take X-ray of the patient, there will be dysostosis multiplex and also bullet shaped middle finger, okay, in the X-ray of the patient. Okay, and also there will be really body inclusion WBC, that is the pathology. So in pathologists will identify this kind of findings. Understand? That is the really body inclusion WBC. Now what are those mucopolysaccharidosis? We have specific diseases name with the enzyme defect that we are going to learn. First is very important, Hurler's disease. Hurler's disease also called as MPS1H. What is this H stands for? Hurler's disease. Okay. Now what is the defective enzyme? Here defective enzyme is IDA gene, but very important, you should remember the what kind of mutation it is, nonsense mutation, and because of this nonsense mutation, with the help of the IDA gene, that will lead to the Hurler's disease. Okay, and the deficient enzyme, that is alpha L iduronate, that is the alpha L iduronidase. Okay, next very important disease is Ske's disease. What is this Keys disease? It is also defective IDA gene. But what is the difference between Hurler's as well as Keys disease? It depends upon the mutation. What mutation, sir? It is missense mutation. Understand? See, this is always, this kind of gene is always active, but here it is, the enzyme is partially active. Which enzyme? Alpha L hydrogenase. See, it is same. The deficient enzyme as well as defective gene is same. It depends upon the mutation. Okay, it differs in the mutation. Here it is nonsense mutation, and here it is, in this case, it is missense mutation. Understand? Okay, so it is partially active and there will be no intellectual disability. So no intellectual disability. So we can find the notings here. Okay. Next, very important is Hunter's disease. Okay, so Hunter's disease. Before that, in Keys disease, it is also called as MPS 1S. Okay, 1S. S stands for Keys disease. Note down. Okay. So next, the Hunter's disease, it is MPS 2. Okay, and the defective enzyme is IDS, and it is a X-linked recessive disorder. Okay, X-linked recessive disorder means mainly the males will be affected. What is a defective enzyme, Amma? Very simple. Alpha L iduronate sulfatase. Alpha L iduronate sulfatase. Okay, and here the very important point, there is no corneal clouding. Very important Amma question. No corneal clouding and only males are affected. That point you know. Okay, so I am repeating again. The defective enzyme is IDS and is a X-linked recessive disorder. And what is the deficient enzyme? Alpha L iduronate sulfatase. And very important, there is no corneal clouding and only the males are affected. Okay. Next, we are going to see about the I, that is the inclusion bodies, inclusion cell disease. What is this, sir? Listen carefully. In inclusion cell disease, it is not a mucopolysaccharidosis. Why, sir? Then why it came here? I am saying it is not a mucopolysaccharidosis. Okay, it is a lysosomal targeting disorder. Then what is mucopolysaccharidosis? Lysosomal storage disorder. Okay, here it is lysosomal targeting disorder. Now listen carefully. We have ribosomes. Okay, and because of this lysosomes, it is producing what? Lysosomal enzymes. And when this lysosomal enzymes, okay, along with the enzyme, that is the mannose 6 phosphatase, it enters to the lysosomes. Here, lysosomes. Now listen carefully. When this is a lysosomal targeting disorder. When there is no M6, that is mannose 6 phosphatase. Yeah, mannose 6 phosphate is not there, then your lysosomal enzymes will not enter into lysosomes. It will be outside. It will be staying outside, is not entering the lysosomes. Understand? And because of this, there is accumulation of the mucopolysaccharides, and because of this, there will be general features for mucopolysaccharidosis, but it's not a mucopolysaccharidosis, sir, because it is a lysosomal targeting disorder. Understand? Very simple. Okay, now we will see here. The defect is in mannose 6 phosphate. We saw now. So there is defect in the mannose 6 phosphate. And what is a deficient enzyme, sir? Very simple. N-acetyl glucose phosphotransferase. N-acetyl glucose phosphotransferase. Okay. Now here, this N-acetyl glucose phosphotransferase, very important to convert the mannose to mannose 6 phosphatase. Understand? Now this mannose is not converted to mannose 6 phosphate because there is a deficient enzyme in the N-acetyl glucose phosphotransferase. Very simple. So mannose is not converted into mannose 6 phosphate, and because of this, our lysosomal enzymes is not entering to lysosomes. Okay. Okay, so the lysosomal enzymes fail to reach the lysosome and MP accumulate in the lysosome. We have discussed. Okay, now what is the investigation? Further on investigation, and what will be the, and we are going to see about the Natowicz syndrome also. Okay, so here in mucopolysaccharidosis we have something called as Sanfilippo. Sanfilippo means mucopolysaccharidosis with no mental retardation. Listen carefully. With no mental retardation, if you have mucopolysaccharidosis, then we call it as what, amma? Then what are the examples? If you see, one is Ske's disease, we have discussed already. Okay, Ske's disease, that is MPS 1S, yes or no? Okay, and also Markowitz disease and also Markowitz-Lamy disease. Okay, these are the examples. Markowitz disease, MPS 4, and also the Markowitz-Lamy disease, that is the MPS 6. Okay, fine. Now, if you look at the investigation of the mucopolysaccharidosis, first we are going to see in the urine analysis and then enzyme deficiency. So no. So whatever we have discussed, we discussed some of the enzyme deficiency. Okay, so we are going to see in the what are the enzymes deficient in the leukocytes, and further on we will be seeing about the genetic mutation studies, and last one, that is the Natowicz syndrome. What is this Natowicz syndrome, sir? The deficient enzyme is hyaluronidase, and the patient will come to you with the clinical features of joint pain as well as the short stature. That's all. So the enzyme deficient is hyaluronidase. That is seen in the Natowicz syndrome. Okay. So this is about the glycosaminoglycans and mucopolysaccharides. Okay. I hope you liked it. Like, subscribe my channel. Thank you. Have a nice day.